In this video, we're going to be showing you how to use the AlphaFold2 Multimer model on the Neurosnap platform. So if you don't already know, the Neurosnap platform is essentially a website where you can access all these different bioinformatic tools and models absolutely for free as an academic and with very user-friendly options for commercial individuals as well. So first things first, we're actually going to go over to the AlphaFold2 service. And then from there, what we're going to do is we're going to input all the different amino acid sequences for the proteins that we want to predict in our complex or in our multimer. So in this particular example, what we're going to do is we're going to take a look at the programmed cell death protein 1 from humans, as this is a very therapeutically important protein. And we're just going to download the amino acid sequence from UniProt and paste it over here. So one thing we do need to do is we need to make sure that the actual header of the FASTA is, in a file format friendly format. So we're just going to clean up the header right there. And then to specify the next chain, what we're going to do is we're going to delimit it using the colon character like so. So in this particular example, we're going to be adding a small peptide binder that we designed using the RF diffusion service on Neurosnap as well. And then from there, what we're going to do is we're just going to go specify some other settings that I think are going to improve the results of this prediction. So a lot of these different settings are already explained in the other AlphaFold tutorial that we have available. In this particular case, I'm going to enable PDB70 because this protein over here already has a structure, and we don't really want to interfere with the structure too much. So this will help AlphaFold2 kind of cheat a little bit by using an existing structure. Additionally, we're going to increase the recycles as increased recycles tend to result in higher quality predictions. And we're going to lower this so that we get all those different recycles unless it's absolutely necessary to stop earlier. Now we're going to run this and we're going to let that run on the Neurosnap servers. And then after that, we're going to come back to you with the results. Alright, so we are now back with the results, and as we can see, this job took about 12 minutes to run, which is pretty standard for a multimer of this size. And the first thing you'll notice is we kind of have like this nice big disordered region. This is pretty standard for membrane-bound receptors, and what these colors indicate is the PLDDT, which is an AlphaFold2 predicted confidence metric that can tell us which part of the structure are most reliable. So the best way to interpret PLDDT is that it confers with AlphaFold's uncertainty in the spatial orientation and position of a particular amino acid. Additionally, another thing to keep in mind is that regions with lower PLDDT, or regions that are more orange, tend to also be more disordered. So in this case, because this is a disordered region, AlphaFold does in fact predict it correctly, even though it's giving it a low PLDDT score. So next up, what we're going to do is we're actually going to color by chain as well so we can see the binder more clearly. And as we can see, the binder does in fact target the main domain that we were targeting with pretty high accuracy, or at the very least pretty high affinity according to the structure. So if we look at the PLDDT again, we can see that the binder structure also is predicted with high confidence, which is exactly what we want to see for an experiment like this. Next up, we have a couple other options that we want to take a look at. So these predicted metrics, we've already covered PLDDT. This is just the average PLDDT. And in this case, we don't have to worry about it being a little bit lower than we'd want it to, just because the vast majority of this protein is disordered. So that's going to have a bias on the mean PLDDT. Additionally, this is also going to impact the PTM score, so we don't really have to worry about it too much in this particular context or in this particular situation. The main score we want to focus on is the IPTM, which is the interface PTM score. Essentially, since the IPTM is very high in this particular case, it tells us that it's a very positive indicator in the reliability of the structure. Additionally, we have some uncertainty metrics that are outputted on Neurosnap and Neurosnap only. So uncertainty is a metric between 0 and 100, and the lower the uncertainty, the better. So as we can see, the uncertainty score for a lot of these proteins is actually quite low. So this is a great thing to see. And additionally, the overall quality can actually be ignored, just because, again, the quality of the structure is being biased by these disordered regions, which we can safely ignore for this experiment. Next up, AlphaFold2 and AlphaFold2 Multimer both output 5 structures each from the 5 different models that were trained by Google's DeepMind team. So we can take a look at all of them just for a little consistency check, and we can see that the structures are overall quite consistent. The peptide kind of binds to the same general region, which is definitely a very positive indicator. Lastly, we have a couple other metrics. So PLDDT, the PLDDT chart is basically the exact same thing as what we saw over here, but instead of being colored by residue, it's just shown, from start to end of the protein. Additionally, the sequence coverage is just the MSA sequence coverage. So AlphaFold2 and AlphaFold2 Multimer receive an input multiple sequence alignment, and this is a visualization of that multiple sequence alignment that was used to predict this structure. Additionally, we can see these PAE charts, which kind of confer nicely with the different domains that we're seeing. So the main domain over here, which is like this blue section, we can see that it's represented nicely in all these different PAE charts in this particular blob over here. And that, this part over here, this little nice cube, is essentially going to be the peptide that we designed to bind to the receptor. And we can see that it interacts very nicely with this region as well. So this is also a great indicator in telling us that AlphaFold is at least very confident in the possibility of this structure happening in real life. So I hope you enjoyed this video, and if you have any questions, definitely leave a comment and we can try to get back to you. And we hope to see you in the next video.